Okay, so good evening, everyone. Welcome to the live interaction session for week 6. So, as we normally do, today we will be first revising the topics for the previous week, and we'll discuss some questions that are related to the topic, some sample questions, which are similar to the questions you have for your assignment as well. And apart from that, we'll also discuss the assignment questions from the last week. And if there are any doubts in that regard, we'll try to figure those out as well. Apart from that, we will have two more sessions in the upcoming weeks. One will be on this Sunday and the other one the upcoming Sunday. Following that, there will be one extra session where we'll wrap up the entire course with all of the materials. I'll give a brief overview of everything that we have discussed. And if there is any doubt from any of the week's contents, then you can certainly bring that up and we'll try to address that. And apart from that, if you have any topics that you feel are relevant for the exam ahead as well, we can discuss those. And with that, we will end the course and complete all of our live interaction sessions. So that is the plan for today. Now, first, I will go through the questions that were a part of week 5 for the topics that were centered around meiosis and the differences that you can see in nuclear organization in each of the different stages and what comes in what comes in that regard as well. So I think it is zoomed out properly. Yeah. So for the previous week's assignment, the first question was about bivalence. We discussed the bivalent structure when it is observed during prophase and how it is a significant distinguishing feature during prophase and what is the significance of it, why is it a structure you only find in meiosis, and what are the implications of formation of a bivalent, how it contributes to recombination, and therefore it brings about genetic variation in the individual or um, initially in the gamete and then in the individual that is formed as a result of fertilization of that gamete, and how that is responsible for adaptation, etc. So coming to the question, it is asking us about the incorrect statement about bivalence. What exactly are those structures? So the first option is that they contain two homologous chromosomes with two sister chromatids. That is true. It is um, like when we talked about recombination, we said that it is an event or a phenomenon that is observed between the non-sister chromatids of two homologous chromosomes. So we had two sets of chromosomes, like two copies of chromosomes, and between each of the two sister chromatids, recombination is not fruitful. So the event occurred between two non-sister chromatids of homologous chromosomes. That structure where you had this overlap or this formation of chiasmata or sites of crossing over, that structure was identified as a bivalent during the process of meiosis. So yeah. So the first statement is certainly true. You have two homologous chromosomes and both their sister chromatids are present. The site of crossing over might not necessarily be between both of the sister chromatids or might not involve both of the sister chromatids, but they are present in the structure. Second option is they are linked together by chiasmata. That is true. They are the points of crossing over. If you remember those structures, the X-like structures that you could observe, or the points of contact that are observed between the non-sister chromatids of homologous chromosomes are referred to as chiasmata. They literally mean the sites of crossing over. Um, Third option is that they appear during zygotine. That is also the correct option. If you remember, we had discussed how the entire prophase can be described as LZPD square, leptine, zygotine, pachytine, and diplotine diokinesis. So among them, during the zygotine stage, you can distinctly identify this particular structure, which is the bivalent, and therefore option 3 is also correct. And so by default, the incorrect option is option 4, that is none of the above. Then in question 2, we were asked which of the following diseases is not a consequence of improper chromosome segregation. Excuse me. So improper chromosomal segregation, or in other words, you can describe it as a disjunction of chromosomes. That is, the proper segregation during anaphase might not occur, or not just anaphase, but at any point of time, if improper distribution of the genetic material does not occur, then that can lead to these diseases. So we have been asked which, please read the question carefully. We are asked that which of these is not a consequence of chromosomal segregation. So we have Alzheimer's, we have Down's, Turner's, and none of the above. So as we have described multiple times, Down syndrome is the consequence of trisomy of 21, which means that there are three copies of chromosome 21 in a particular individual, which occurs due to improper duplication as well as improper segregation that has occurred, that both of the daughter cells from a parent cell an equal are not getting equal genetic content at the time of cell division, and that has led to this imbalance. So therefore uh, Down syndrome is certainly a disease that is observed, and we also had discussed how it is often linked to even the age of the mother. That is, over time, you can see that there is a weakening of the microtubules that are present within the ovum. And as a result of that, there can be some sort of a anomaly due to which the microtubules are not participating correctly during the process of pulling apart of the two sister chromatids during anaphase. So that is certainly a consequence of improper chromosomal segregation. The other option is Turner syndrome. Now, during Turner's, a similar event occurs, but it is a little earlier. It is during the process of during prophase itself, that when the gametes are formed, or in fact, it can occur sometimes during meiosis two as well, during the second stage of meiosis as well. So the gamete which is getting formed is null for one particular gamete, one particular copy of the chromosome, which is why the phenotype of the individual is different, and the genotype is recognized as XO. That means they do not have two copies of the X chromosome. So Turner's as well as Down syndrome are both a consequence of improper chromosomal segregation. However, Alzheimer's disease is not the result of that. Even if you're not very familiar with Alzheimer's, it is it is a neurodegenerative disease which occurs due to the formation of certain structures within the neurons, which are if I put it a little in a layman language, it occurs due to the accumulation of one particular protein, tau protein, within the brain, which it aggregates inside the brain. So that protein gets phosphorylated, which means a phosphate group is added to it. And as a result of that, it is able to form very haphazard structures in the brain. They are often known as neurofibrillary tangles and plaques. So you might have heard of dental plaque, that it is sort of a structure that builds up over the teeth of individuals who do not go through regular cleansing for a long period of time. So that is a similar sort of biofilm-like structure. It's not a biofilm. I'm just trying to draw the analogy from biofilms, but it is somewhat of a 3D structure which is often observed within the brain, and that leads to progressive death of neurons. Now, because neurons are not constantly dividing, they are said to be end differentiated, which means that you have a certain number of neurons that that you're born with and that develop till the age of 4 or 5, and beyond that, you do not have a further division of neurons. The only thing that happens is that new synapses are formed, or new connections between neurons are formed every time you exercise a certain activity, which is why when you memorize something, you're able to remember it because a particular synapse has been activated in that regard. Now, when such tau protein accumulation occurs, it leads to death of these neurons. They are not able to survive. And as a result of that, these connections, these synapses that I talked about, they are lost over time. So one of the major phenotypes that are observed in neurodegenerative diseases, such as Alzheimer's, as mentioned, is the loss of memory, otherwise known as amnesia. So that is the first one of the preliminary phenotypes that you're often able to observe. There are other neurodegenerative diseases as well, which have different causative backgrounds. Like, for example, there is Huntington's, that is different. Dementia by itself is a little different. And of course, there's a whole area of research that goes into it, but these are not direct consequences of chromosomal segregation. Therefore, the incorrect option in this regard would be Alzheimer's. So following that, in question 3, we were asked that the Saccharomyces cerevisiae IME2 promotes entry into which of these following phases. If you remember, it was a part of the lecture notes directly. So mitotic S phase, meiotic S phase, or the meiotic G1 phase, or the mitotic G1 phase. So IME2 in yeast, Saccharomyces cerevisiae, promotes entry into the S phase of meiosis. It operates at the checkpoint, which ensures that a cell has now committed to the process of replication, and then it will eventually proceed towards cell division. Then in question 4, we were asked that classical NLS, or the nuclear localization signal, is recognized by which of the following? Importance, exportance, lamina, or all of the above. So if you remember the entire structure of the nucleus that we talked about, and how transport into and outside of the nucleus is being executed by this group of proteins, then the recognition of NLS, because NLS, even if you don't completely know the answer, if you just go by the name of this particular sequence, which is the nuclear localization sequence, it means that it will ensure it acts as the address for a protein, that this protein is supposed to reside within the nucleus. So its localization or its transport would be carried out by the group of proteins that operate in this regard. So in this case, that would obviously be the importance, which are importing proteins from the outside, or outside basically means the cytosol after synthesis has been completed by the ribosome. From the cytosol, it has to be localized into the nucleus. So importants are carrying out this job, and for executing that job, they will first recognize the NLS that is present mostly within the N-terminal sequence of the polypeptide chain that it is currently looking at. So that would be the correct option. Exportants are responsible for nuclear exclusion to be transported outside of the nucleus, and the nuclear lamina is part of the nuclear envelope, which is responsible for segregating the contents within the nucleus from the cytoplasm. So that does not necessarily perform this job of recognition of NNS. Um, then coming to question 5, again, please interrupt me at any point of time and ask if you have any doubts regarding my explanation or regarding the question itself. Just a moment. So in question 5, we are asked that you need to pick the incorrect statement about RAN. Uh, First, it is a monomeric GTP binding and hydrolyzing protein. It is that it is a it undergoes this GTP-GTP exchange. It has the GTPase activity, by virtue of which it is able to do that. And that exchange is crucial to the process of import and export from the nucleus. It is one of the main regulators that is executing this entire transport in and out of the nucleus. Second is that RAN works with importance. It does. It is responsible for the import of proteins. RAN can exist in two different conformational states. Certainly, yes, like we said, it is executing this switch between GTP and GTP. And fourth option is that RAN is an ATPase. So that is the incorrect option, certainly in this regard, because it is the energy molecule that it is associated with is GTP, and it is responsible for hydrolyzing GTP, and that exchange is mediated via its transport only. So ATPase is certainly not the activity that RAN upholds. So that would be the incorrect option. Uh, then coming to question 6, the nuclear export signals are likely to be rich in which of the following? Leucine, serine, threonine, or histidine. If you remember, the nuclear localization signals are usually lysine rich, lysine arginine rich, but the export signals are usually found to be very leucine rich. So when we say leucine rich, it means that if the sequence is of, say, 20 nucleotides, for example, 20 amino acids, for example, then a good percentage of it, say 70-80% of it, would be very leucine rich, and there would be a few other amino acids. And the reason why that is relevant is because the presence of such um, a sudden increase in the abundance of a particular amino acid within a stretch of a few or within a short sequence is what makes it recognizable to the machinery. So if you see that for a protein to have a stretch of leucines at the beginning or at the end, at the N or C terminus, then it becomes very easy for the recognition molecule, for the exportin in this case, to identify that this is a protein that has to be moved outside, that needs to be shipped outside. Versus if it has this particular lysine rich motif or arginine rich motif, then that also becomes easy for the protein to recognize it and then carry out whatever job is necessary. So the abundance of an amino acid is sufficient to transport it to its proper destination. So that was the question number 6. Then in question 7, we were asked pairing of homologous chromosomes and crossover occurs during which phase of meiosis? So this entire process of the LZPD square that I talked about, all of that takes place during the prophase of meiosis 1, otherwise known as prophase 1. So that would be the correct option. Of course, not during metaphase, anaphase, or telophase, where we know that there is alignment of the chromosomes, separation of the chromosomes, and then rebuilding of the nuclear envelope in the subsequent stages. Then that would be followed by meiosis 2. So that these are the distinct features of each of these stages. So prophase 1 would be the correct option. Um, which of the following is a meiosis-specific kinase? IME1, IME2, REC8, or SPO11? So if you remember, we just, in one of the previous questions as well, we saw that IME2 is what allows that entry into the replicative phase of meiotic cell division, and therefore IME2 would be the meiosis-specific kinase in this case. Then in question 9, we were asked which of the following nuclear bodies plays a role in the processing and storage of SNRNPs, PML bodies, polycomb bodies, canal bodies, or none of the above. I had briefly introduced the different types of nuclear bodies that are also present, and it was even present in the lecture notes by Professor Laloraya as well. So Kahal bodies are the answer to that. They are responsible for the small nuclear RNP processing as well as storage with inside the nucleus. So Kahal bodies. Again, this is a very direct question, so I'm not elaborating on it much, but Kahal bodies were the answer to it. Finally, in question 10, we were asked that during which phase of meiosis does the clustering of telomere occur? Again, this was one of the distinguishing features that we identified in each stage, and there were multiple images as well in the lecture notes and probably in my slides as well, where we saw how clustering of telomeres can be identified during prophase, and particularly within prophase during the zygotane stage, where you can see the formation of bivalents occur. So clustering of telomeres occurs during the zygotane stage. Therefore, that would be the correct option. So with this, the assignment for the previous week was complete. And I hope you were able to get most of the questions right, if not all. And if there are any doubts, you can certainly go ahead and ask. And then we'll go through a brief recap of the topics covered in week 6. So please let me know if you have any questions. Okay, if not, you can ask at some point later during the class as well. Um, for this particular week, the topics were majorly centered around chromosome organization, and I think we have briefly discussed it previously as well, but this week got into a lot more detail about it. One thing I'll say in advance is that the later 2 lectures, we had 4 lectures for this week. So the topics of SMC protein and the topic of how it is organized and how it is studied has been taught by some of the biggest experts in the field. So, and that is the most up-to-date information you'll find as well in this regard, because they are active researchers in this field as well. So I have not put a lot of detail regarding it. I have focused a little bit on the technique, very briefly on the technique. If there are any doubts, I'll certainly try to answer those. But additional material in that regard at this moment is not something that I have. What I'll focus more on is the cell biology aspect of it, the um, not just even cell biology, the epigenetic aspect of it as well, because the entire organization of chromosomes is a topic that is breached between both cell biology and epigenetic organization of or epigenetic modification of the genetic material within a cell. So that is the domain I'll be focusing on more for this week. Regardless, if you have any questions, we can certainly address those. And if not, I'll come up with more material to help you in the next 3 sessions that we'll be having. So with that in mind, the first thing that I would like to get your attention to is the fact that, okay, so the book that I have majorly referred to in this regard is the book by B.M. Turner. I have cited it at multiple places. You'll find it at the bottom as well. So this book is, while slightly old. If you have any access to a library, you will get better updated versions of it. The PDF is slightly old, but it has the most beautifully described information regarding how genetic material, how chromatin is organized inside the cell. So if you want to go to the basics, this would be one of the best books that you can refer to. There is also a book by, I think, Gardner. Its PDF is also difficult to avail. If you have access to it, you can go ahead and refer to that as well. Otherwise, you will certainly find open access papers online. You can refer to PubMed. You will get a lot of information there. And you'll find review papers also. If you're somebody who is in the habit of reading papers, then that is something which would be good for you. And it would give you a more comprehensive view of what the initial starting point was and what is known today, and how this entire timeline has evolved with whatever knowledge we have gotten in the past several years, several decades, in fact. So, with that in mind, let us first understand what the focus is for this particular week. So, the first images that you're seeing are majorly from that book only. So, what was done? What was the first? One of the first ideas that brought about the idea of chromatin organization, that something like that has to happen, came from the fact that uh, whenever we talk about the genome, we understand how huge it is, and we understand that the dimension of a cell is too tiny to accommodate such a huge genome. So the easiest analogy that you can draw is, for example, intestines, right? We know that the small intestine is so long, it's several meters long, right? But compared to that, as individuals, we are obviously much smaller than the length that the intestine covers. So the way that the body accommodates the small intestine, I think it's approximately 7 feet or something. So 7 meters, sorry, 7 meters. So that huge or that extensive area is packaged into a small space because of the twisting and turning and compaction that it is subjected to. So in order to concentrate the area that it is taking into a very small space. So taking that analogy to what is happening within the nucleus or for the genome inside the cell, we see an extensive amount of compaction that is occurring. Now, the compaction is not just a space-saving strategy. Certainly, that is relevant to the cell. In order to accommodate the genome, it has to undergo such extensive compaction. But that is not the only purpose. It is necessary for a number of other things. One easiest example for it would be DNA damage repair, which is something that came up in the later lectures as well. So that is the primary purpose of it, is to accommodate the genome. But the secondary purpose, and rather the more important or more energetically relevant purpose of compaction, is in order to make sure that the cell does not aberrantly express anything and everything at any point of time. So the major control of gene expression that occurs within a cell is maintained at the level of compaction, at the level of nucleosomes, which is something we'll come to at this point of time, just right after I complete this part. So the accessibility to DNA that is provided within a cell can control what will be expressed and what will not be expressed inside a cell. Because DNA by itself is not dynamic in nature. DNA is a stretch of nucleotides that are present within a cell. The control is occurring either above DNA, which is where epigenetic concept comes in. Epigenetic literally means above genetics. So something is being done to the genetic information. In some way, it is getting modified in order to control the expression of a certain gene within the cell. So this control or this fine tuning that we talk about inside a cell is crucial for its survival. Whenever this is not being obeyed, it will affect the cell health. It can lead to cell death, and it can have far-reaching consequences, which can lead to a number of diseases. So the correct control and the correct regulation of genetic expression starts from this very position, starts from the epigenetic modification, starts from the condensation or the accessibility that is given to the genome at any point of time. Apart from this, of course, it is relevant in other ways as well. Like we have understood that during the process of meiosis, it is required that it has to be condensed into chromosomes. Then it again undergoes decondensation. All of that occurs based on what phase of its life or what phase of the cell cycle that particular cell is in at that point of time. But The far-reaching impact remains that the control of genetic expression is maintained by this compaction and condensation, and therefore it has to be understood and has to be understood thoroughly. So what exactly is happening inside a cell? It was initially observed by scientists, if you look here, that when the DNA that was present was hybridized to mRNA. You can see that there is hexon mRNA and there is adenoviral DNA. When you have imagine two strings, if you have one string which is longer, which is your DNA, and to that you are hybridizing something which is the RNA for the same, which is slightly shorter, you'll be able to see that it is not perfectly hybridizing with each other. You can see that there are certain regions which are open or which are not completely hybridizing. What are those regions? Those are usually introns. Now the question might come up that if there is a longer DNA, why is the RNA not the exact same length? Why is not all of it transcribed? Why is some of it not accessible? Whoever has studied transcription knows that as part of post-transcriptional processing, splicing occurs, right? And some part of DNA is, even though it is transcribed, it can be removed or it might not get transcribed at all. All of these are processing steps that have been brought in order to fine-tune the entire process of transcription, especially in eukaryotes. It's not a not a prokaryotic measure that you can see. In eukaryotes, you can find this specific event occurring. Now, this was one of the key experiments that were performed a very long time ago by scientists, and the impact of this experiment was something that people understood in the long run. So I'll just read it out. So the discovery of splicing was followed by several other experiments with cellular mRNA, which showed that the eukaryotic genome had a very unusual structure. Why I brought this up was because while splicing was something that was being studied by a whole other group of scientists, it was the first indication that what you expect to see inside a cell, that the genome is simply present, is not the true case. The genome has a lot of modifications, a lot of finesse to its arrangement inside the cell, and this was one of the preliminary indications of it. So rather than being continuous, their coding sequences are interrupted by introns, which are removed by splicing. Introns now, of course, we know they account for a huge chunk of the entire eukaryotic genome, and the role of introns in evolution and regulation of gene expression are still areas of active research. The discovery of splicing also stimulated interest in the mechanism of unexpected RNA processing, which is something we are not getting into right now. But this unexpected structure had a major impact on cellular and molecular biology. So this was one of the, like I said, one of the preliminary indications that the genome that is present inside the cell and the resultant RNA or the transcriptome that is present inside the cell can be uh, completely different, or well, of course, they are not absolutely different from each other, but the extent of expression that you can see are very, very different from what you expect. Not every genetic information inside the cell is expressed, nor is it expressed at all times during the life of a cell. So there is this particular terminology that is used with respect to gene expression that is called as spatiotemporal regulation of gene expression. Spatiotemporal literally means the space and the time. So the genome that is present inside a cell is the same for every cell within the living body. Of course, there are exceptions to it. For example, RBCs which do not carry a genome. So keeping those aside, every other cell which is part of a functional aspect of the body, which is part of a tissue that is eventually forming an organ and performing a certain function, all of them have the same genetic material. But the control of expression of that genetic material is maintained both with respect to the space and with respect to the time at which it is occurring. What this means is that when we say space, it means that despite the presence of the same genetic material in all tissues, expression of a particular gene can be tissue specific. For example, if there is a certain gene which has to be only expressed within the heart, then its expression will be controlled by the living organism, by the individual, in a way that the expression of the gene is only restricted to the heart and does not occur in the liver, does not occur in the brain or somewhere else. This haphazard expression leads to problems, leads to a lot of difficulties, let's put it that way. For example, oncogene expression leads to cancer, or the absence of expression of certain genes, for example, when we talk about tumor suppressors, can also lead to cancer. So these space-based or the tissue-based expression is something that regulates genetic expression within an individual to a large regard. The second aspect of it is the time. So despite the presence of the same genetic material, even within the same tissue, you can see that not all genes or not all the tissue specific genes are expressed throughout the life of an individual. The very simple example of it is cell cycle that we have so extensively studied, right? So cycling genes, which have that cyclical expression that we talked about so extensively, is something that is being controlled with respect to the time. What phase is the what phase is the cell currently in? Is it facing some specific stress? For example, if a cell has to enter autophagy based on whatever stressful conditions it is facing, then at that point of time, only a particular group of cells which are facing stress within the body, within a particular tissue, within a particular subtype of the tissue, only will the expression of those stress genes occur. Not all of the cells within the body of an individual will die. So that specific expression and that specific regulation is what is very crucial towards the survival and the proper setting of an organism and its proper uh, like proper survival despite whatever conditions it is facing. So this genetic expression is being controlled at multiple levels, not just epigenetic, genetic, or transcription, but at post-transcriptional, post-translational levels. So there are multiple checkpoints that are in place. Right now, we are talking about epigenetics. So when we say epigenetics, like I said, it means control at the level above the gene. So this control is being regulated by the packaging of the genetic material or packaging of DNA into these distinctive structures that you already are familiar with, which are nucleosomes. So if you look simply, I think there is this image. Okay, it's not there. Okay. Anyway, this should also be clear. So what you see here on the upper right is the structure of nucleosomes. What you basically see is the beads on string structure. What that means is if you have sort of a, you must have come across it at some point of time. If you see any necklace, which usually has little beads that are attached onto a string, that is what DNA can look like if you look at it under an electron microscope when it is not condensed into chromatid. That is because of the presence of these nucleosomes. Now, these nucleosomes are basically a protein moiety which is surrounded or which is wrapped around by DNA. One of the analogies that I very commonly use is the computer mouse. I think it's the easiest example to understand. I think I might have introduced it before also. So if this is your histone octamer, the DNA that is wrapped around it is what is forming the nucleosome. Now, again, this is getting extended. There is a linker region. Following that, there is another nucleosome and so on and so forth. So this is how your entire, that huge extensive DNA is undergoing its first level of compaction, and you are reducing the space and the area that it will take inside the cell nucleus. Now, what is the octamer formed of? It is made up of one particular group of proteins, which are known as histones. And these histones, which are particularly present inside the nucleosome, they are H2A, H2B, H3, and H4. And after that, after one particular turn has been formed, you have the histone octamer around which approximately 147 base pairs of DNA are usually bound, following which there is a linker region. Now, to demarcate that this is the linker region, there has to be a stop. If you think of, for example, your door has a stopper, or when you tie a balloon and you tie a knot on top of it, so that it limits the area within which that is present. That stop is the histone H1. So while histone H2A, H2B, H3, and H4 are part of the core histone proteins or the histone octamer, H1 is present a little bit on the outside, and it marks as the stop between the nucleus between the nucleosome and the linker DNA. Now, this has some flexibility, and that flexibility is something that has been identified later, and that has been discussed also as a phenomenon known as nucleosome breathing. So again, let me use this reference only. I think it's the easiest way to explain that. So if you have this, just a second. Right. So if this is your histone octamer, sorry, around which your entire DNA is undergoing almost two turns approximately, and then you have this extended region, and you have your linker DNA stop here, right? Despite the presence of the linker DNA stop, that is histone H1, there is some flexibility, as in the DNA can move a little on either side so as to release that stretch of DNA which is now accessible to the transcriptional machinery. So this phenomenon where the nucleosome can move a little bit, very slightly, it's not a huge movement because it will be an energetically very, very expensive process. There is some amount, a few base pairs that can be exposed on either end, and that sort of shifting movement is known as nucleosomal breathing. Now, this is not enough to change the expression of a particular gene, because that can have very huge impact or huge consequences on the survival of the cell. Therefore, when it comes to the expression of a gene, then it involves other proteins, mostly which are known as chromatin remodelers, to come in. They come and they remodel chromatin. It means that they are able to either remove these histones, or they're able to move them on either side, or they can modify these histones, all of which can lead to unwinding or winding of DNA. And therefore, it can control whether that particular region of the genome is now free. When I say free, it means that transcriptional machinery can come and sit on it and can transcribe it, convert it to mRNA, then generate the protein from it. Or if it is not free, which means it is not accessible. So the major control, again, I'm repeating the same thing, the major control at the epigenetic level is by ensuring that a particular region of the genome is accessible or not accessible. If it is accessible, then that region of the genome is said to be the euchromatin. It is accessible or it is open chromatin. If it is not accessible, if it is bound by proteins, mainly by nucleosomal proteins or histones, or by any other proteins, by any sort of a protein which is imposing some sort of a block. In other words, if it is imposing a steric hindrance, that there is something present, which is why the transcription machinery is not able to go there, then that region is inaccessible, or it is closed chromatin, or it is otherwise known as heterochromatin. So these are two major regions of the genome. Euchromatin is open, accessible, it is transcribed or transcriptionally active, or heterochromatin is bound by proteins, it is closed, it is inaccessible, and it is not transcribed, or it is silent. You can say silent or inaccessible. These are all interchangeably used. Now, whatever modifications are being done are being performed in order to change whether or not that particular region of the genome can be accessed or not accessed. That will control whether that region gets expressed or does not express. So these are all coming one after the other. I hope it is clear to you. But that is the far-reaching purpose of all these steps of genetic and epigenetic regulation. So what you see here is the chromatosome. The chromatosome is the structure which involves the core histone along with the DNA which is wrapped around it, and histone H1. This entire entity is chromatosome. Chromatosome without histone H1 is the nucleosome. So these two terms are different, but just for your understanding, I mentioned both of them. So the image which was at the bottom right is also another cartoon representation of the same thing only. And that is basically what constitutes a nucleosome. What you see here is the structure or the domain organization of histones. So histones, like we said, majorly these four types of histones are present as part of the octamer: H3, H4, H2A, and H2B. What are these made of? You can see that they are approximately all around 140 amino acids long polypeptides, and they majorly have three alpha helical domains that are present, which are labeled as alpha 1, alpha 2, and alpha 3. And you can also see that they have this conserved organization is present in all four types of histones. They, of course, have sequence variations, and the position of the alpha helices is also different. But the conserved idea between all four of them is the presence of these alpha helices. What is the necessity of it? The necessity lies in the fact that the presence of these histones, because um, understand the fact that there are 8 proteins, and when the job of 8 proteins is to compact DNA, what needs to be understood at a very basic level is that these 8 proteins must also be very, very closely compacted with respect to each other. So this close compaction is brought about because of a certain interaction that in common terms is understood as a histone handshake, which means that the presence of three alpha, if you look at the diagram on the right, you'll be able to understand it better. It shows the histone handshake between histone H3 and H4. So you can see that the alpha helices, they orient themselves in a particular manner such that they are fitting most, like obviously I'm not able to show it by my hand, but the fit between them is so perfect and so complementary that they are able to interact at multiple locations. Particularly the helix-helix interaction is such that they are able to sit in perfect alignment with each other and prepare a concentrated octamer around which DNA gets wrapped, and it further condenses or further compacts the genome into a very small space. So the organization of the histone and the interactions that you can see between each of these histone motifs is what brings about the first level of compaction within the genome. And what that effectively forms is known as the 30 nanometer fiber. So from the DNA that was completely open, it is now condensing to a region which has a thickness of 30 nanometers. What you see at the bottom, again, these are all images from the book by B.M. Turner only. You can go through it. Uh, Excellent book and will clear up your basic concepts. And you'll find a lot of experiments also mentioned there to um, give you a better perspective as to how people understood what exactly is present in the genome. Today, we are understanding it as a fact that this is how the genome is organized. But there is a bunch of experiments that have come that have led to that conclusion to understand that the arrangement has been understood because of these experiments, XYZ experiments. So you can go back and look at those experiments. They're very simple, very basic, but they're, of course, remarkable conclusions that have been drawn from it. And we understand the genome today because of basic experiments performed back in the day like this. Now, one key enzyme that is used in most of these experiments and most of these studies is MNase, otherwise known as micrococcal nuclease. Micrococcal nuclease has a specific way of working. It is used till date in a certain subtype of experiments, particularly if you've gone through the video lecture. You must have seen the 3C assay, the chromatin conformation compaction assay. There, the enzyme which is used to digest DNA, one of the enzymes used to digest DNA is micrococcal nuclease, and it has this peculiar way of working where it surrounds DNA and it cuts DNA in a way such that whenever protein is present, it is not able to execute its cut, but MNase is able to sit on it perfectly and is able to generate that double-stranded cut. So MNase has a particular way of working such that it does not digest the nucleosome protected region, but the free regions that we talked about, which are part of the linker DNA, they get digested. So that is a way of studying how the genome is organized within the cell. Now, coming back to what we talked about, the 30 nanometer fiber, what you see here on the left is the multiple levels of organization that you can see inside a cell. So the first step is the DNA as you know it. The 2 nanometer thick DNA is the DNA double helix that we have all learned till date. And that is what you understand as the genetic composition inside a cell. Then what that effectively forms is the beads on a string structure, which is just simply the DNA and um, the nucleosome, which is wrapped around, or the histone octamer around which DNA is wrapped to form the nucleosome, and that forms the 11 nanometer fiber. Following that, what we see is the 30 nanometer fiber that I was talking about. Now, this is the consequence of the formation of nucleosomes because now these regions are able to pack against each other. So what you saw initially in the beads on a string structure are now able to further concentrate. So what you saw as part of that mouse, multiple such entities are interacting with each other, and this extensive DNA is getting packaged into a much smaller space. From there, what you see is the 300 nanometer fiber, which loops, and then you have the condensed section of a chromosome, which has approximate dimension of about 700 nanometers. And finally, from the entire mitotic chromosome, which gets concentrated into approximately 1400 nanometers, you can see the extensive stages at which packaging has occurred. So the chromosome that you observe under the microscope simply by taking an onion root tip cell and the DNA that you learned about back in the day, there is a huge gap that is present between the two. Not a gap, but a huge level of um, organization that has occurred to arrive from this simple DNA to the chromosome that you're able to finally observe using a microscope. So these levels of packaging are all mediated simply by interactions, by arrangements, and by protein DNA involvement or protein DNA crosstalk that is allowing us to condense that extensive genome into a very small space. Now, uh, if you look at the image on the right, you'll be able to see that we talked about histone H1, right? So what H1 is doing, apart from just being the stopper for the chromosome, it is also bringing about some organization into the previously unorganized DNA. So in other words, you can say that it is um, if you understand the concept of entropy, you will see that a system by default always will try to minimize entropy, which means it will try to bring in more and more organization, bring in more and more order inside the system. So H1, the recruitment of H1 is already compacting DNA because it is coming, these two nucleosomes are coming close to each other. When there is further interaction in a way which we will discuss in the upcoming slide, such that if you look at the figure on the bottom right, you'll be able to see that each of these nucleosomes and their histone H1 are all coming into close contact with each other, and they are sort of twisting around each other to get closer and closer in a way such that the nucleosomes are now close and DNA is now condensed into a smaller space, and there you can see the emergence of the 30 nanometer fiber. This entire process is crucial in order to arrive at the condensed structure that we have been talking about for so long. So how does this extensive organization occur? So what you see in the upper left is the zigzag arrangement and the solenoidal arrangement of the extended chromatin. So you can see there has been numbering which has been made on the nucleosomes also for your better understanding. So 1, 2, 3, 4, 5, and 6. What you see is the arrow indicates the direction in which DNA is getting wrapped around the nucleosome, right? So you start with nucleosome 1. DNA takes approximately 2 turns. Then for the second nucleosome, you have a twist. From the second to the third nucleosome, there is another twist, and so on and so forth. So each of these nucleosomes are packaging in a twisted manner with respect to each other. The other possibility within the zigzag arrangement of nucleosomes is that it might not get twisted, and there is a simple unidirectionality in which the nucleosomes are getting packaged with respect to each other. And this is something that has been observed using electron microscopy, that there is a difference in the way in which nucleosomes can get packaged with respect to each other. Either it can be a simple alignment one after the other, or it can undergo this twist that we talked about, which leads to more and more compaction, and which makes that twisted area even less accessible to the transcriptional machinery or any machinery for that fact. It is becoming a more condensed, more compact, and more inaccessible region. So that is one arrangement, the zigzag arrangement. The other is a solenoidal arrangement of the 30 nanometer fiber. You can see the representation of it on the right as well in blue. So in zigzag, you are able to see that there is a sort of a twist which is occurring at each level, versus when there is a solenoid, you know that a solenoid is similar to a spring to some extent, much like a rosette or a spring, where you have this arrangement, one like the circular arrangements one after the other, and that is a different way of packaging. Between these two, the zigzag one has a more tight packaging, but effectively both of them lead to the 30 nanometer fiber generation. So from the 30 nanometer fiber, like we saw in the previous slide, you will see more and more yeah, you'll see more and more extensive packaging. You'll see that between these solenoids, you will see these looping of DNA, which will occur against a scaffold. If you remember, we had talked about the synaptonemal complex formation, which takes place during prophase 1 as well. So there you will have a scaffold against which this 30 nanometer fiber will form loops. And those loops also have some flexibility depending on which their transcription may or may not occur, and accessibility of the transcriptional machinery will also vary. And further, it will eventually lead to the formation of the chromosome that we know all too well together. So this was the basic condensation and basic compaction that is understood at the epigenetic level for the genetic material inside the cell. If there are any doubts, please ask, and then we'll move on to the modifications, or particularly the epigenetic modifications that occur within the genetic material. Okay, if not, at any point you can raise your hand or interrupt and ask me also. If I'm going too fast, please let me know in that case as well. Uh, okay, so coming back to the slides. So this was the basic arrangement of the genetic material, following which we are now looking at the next level of regulation. Apart from compaction, the next level of regulation occurs with respect to modifications. Now, when we say modification, what does it mean? It means that there is the introduction of some subgroup or some chemical entity onto the existing genetic material. When I say genetic material in this regard, it does not mean that modifications only occur in the DNA. More extensive and more relevant with respect to epigenetic modifications are modifications within the histones. So the histone proteins that we talked about, they are very, very rich in lysine and arginine. In particular, the histone proteins, even though they are so well packaged and they are forming that perfect fit with each other, despite that, what you need to keep in mind is that they are polypeptide sequences at the end of the day. They are not forming little spheres that are all perfectly arranged with respect to each other. No, they have a 3D conformation within which the alpha helices are interacting well and interacting closely with each other. But there are tails that are present, or extended regions, either N-terminal tails or C-terminal tails, which protrude out from the nucleosome. So if you have that entire structure, from there, you will be able to see these little tails that are projecting out of the nucleosome. Even though there is extensive packaging of the DNA, histone tails are found to protrude out or sort of pierce through the nucleosomal structure. Why is this relevant? Histone tails are one of the key sites of modification within uh, the epigenetic realm that we are talking about so extensively. And modification of the histone can have very far-reaching consequences. Why? We'll come to that. Let us first understand the basic modifications that occur. So there are, oh, sorry. So there are mainly two types of modifications. There are more, but mainly two types of modifications that are very, very relevant with respect to gene expression. One is the acetylation, and second is the addition of a methyl group. What you need to understand is that these are not the only two modifications. There are more. Most commonly, you will have histone phosphorylation, you will have histone sumoylation, where a sumoyl group is added, and you can also have ubiquitination. Now, despite the fact that we have understood ubiquitination to be a degrading, degrading mark, that is not its only job. The number, if you remember, I think I had mentioned it in the live interaction session as well, that the presence of a polyubiquitin stretch is serving as a degradation mark, that when there are multiple ubiquitins that have attached, that will be recognized by the proteasomal machinery as a degrading mark, and only then will the machinery be recruited there. But if there is a monoubiquitination or a diubiquitination, then those have different implications, and those are simply modifications, but they're not targeting that protein for degradation. So there is a difference in the number of entities, in the number of ubiquitins that are attached. It can have different consequences. What we are particularly focusing on here are acetylation and, of course, methylation. Why are they so important? So if you look at the diagram here, you will see that you see proteins, and you see two types of enzymes that are operating in switching these proteins from one state to another. What you see protruding from the protein is the side chain group, which is sort of projecting outside. Now, when there is acetyl group addition, that acetyl group is added from something which you are mostly probably familiar with, that is acetyl coenzyme A, which is a molecule that is found to be present extensively within the cell, and that has a lot of implications in cellular energetics and cellular metabolism. But it also serves as a donor of the acetyl group. Now, when acetyl coenzyme A is present, then acetyl transferases or histone acetyl transferases, otherwise known as HATs, which is what you see written below the top arrow. These HATs take the acetyl group from acetyl coenzyme A and add it onto the protein side chain, histone protein side chain, and they are acetylating histone. What does that do? Why is it at all important? The addition of an acetyl group, if you see here, it is indicated as C double bond OCH3, but acetyl group otherwise can, it carries a negative charge, as a result of which it is able to change the interaction that is present within DNA and histone. If you remember, the background or the backbone of DNA in particular carries a negative charge because of the phosphodiester bond that is forming the backbone of DNA. If we go back to the basics, you have each of these individual nucleotides, deoxynucleotides. They are getting attached to each other by the phosphodiester linkage or phosphodiester backbone. Now, the presence of this phosphodiester linkage is rendering a negative charge to DNA by default. On the other hand, histone proteins or the histone octamer that we talked about is very rich in lysine and arginine, like I have mentioned multiple times, right? Lysine and arginine are positively charged amino acids. So apart from the compaction that is present within the histone octamer, the difference in charge between histones and DNA is what makes that compaction so intense, so strong, and so persistent throughout the life cycle of a cell. So the negatively charged DNA is able to wrap around the positively charged protein, which is histone, and therefore that compaction is maintained because of this charge difference or the attraction between the positive and the negative charges. However, when histone proteins, which, like I said, are destined to normally carry a positive charge, when they get acetylated by this histone acetyl transferase or HAT, what that is doing is it is lessening the positive charge which is present on the histone, as a result of which the negatively charged DNA is no longer able to bind as tightly onto the now negatively charged histone, and therefore the interaction between them becomes weak, or DNA in that region is no longer tightly wrapped, and therefore DNA in that region is a little loose. So the acetylation of histones is reducing the interaction between DNA and histone, and therefore DNA is now opening up. So acetylation is considered to be an opening sort of mark, which is rendering that region now to become transcriptionally active, and therefore acetylation is also called as an activation mark. I hope it was clear. I'll just go through the flowchart once again. DNA is negatively charged. It is binding to positively charged histone, and that interaction is maintained very strongly. When this histone is getting modified by HATs or histone acetyl transferases, as a result of which its positive charge is now less, or instead it is carrying a negative charge, then DNA is no longer able to bind to it as strongly, and therefore it will open up, or it will no longer be inaccessible. As a result of this, acetylated histone is now no longer able to bind as tightly as to DNA. What you need to keep in mind is that DNA does not completely unwrap. That does not happen. Only in a localized region, within a particular region, you will see that DNA is no longer binding as tightly as before. Therefore, that region can now be accessed by any machinery within the cell, particularly the transcriptional machinery, and as a result, that region can undergo or can now be expressed, and therefore the acetylation is serving as an activation mark inside the cell. I hope it was clear. If not, please tell me. I'll explain it again and I'll try to explain it better as well. So that is the job of an acetylation mark. On the other hand, when a particular region needs to be closed up or needs to be now made inaccessible, then that acetylation mark from histone needs to be removed. Therefore, this change is not permanent. This change is reversible. Or you can also consider it to be transient, which means that it is only occurring for a particular duration. This again maintains the spatio-temporal expression of that gene, that only within a certain tissue, only at a certain time, you are allowing that gene to be expressed. So this removal of the acetyl group is carried out by HDACs or histone deacetylases, which are removing the acetyl from the histone protein. And therefore, the histone is now again positively charged and it is able to bind to DNA just as strongly as it was before. And therefore, the activation mark that was previously present is now being removed. So this is the level of control that we can see for histone modification in an activating manner. On the other hand, the second type of modification that I talked about, which was methylation, is a repressive mark. As you can understand by the name, it is completely in contrast to the job that acetyl groups do. Instead, methyl group, or simply the addition of a CH3 group, can usually, it is again something that can be debated, but usually methylation will serve as a repressive mark. So what happens in those cases is that donors such as SAM or S-adenosyl methionine will donate the methyl group, which will be carried over by methyl transferases, and that will be added on to histones. Then that can lead to a repression or will not allow that region to undergo expression. However, what you need to keep in mind is that methylation, what we are discussing here, is only with respect to histones. Methylation of DNA is a whole other story, and it is a field which is still being actively researched, and the methylation of DNA is being used extensively in genomics as well. There is a set of genomics research which looks at methylated and unmethylated, hypomethylated, hypermethylated DNA. So that's a whole other area of research. We are talking about histones here. Don't get confused. DNA methylation can have very different and very far-reaching consequences as well. Apart from that, even within histones, at specific residues, the number of methyl groups that get attached can also have different consequences. So histone monomethylation, dimethylation, and trimethylation can have different marks. It simply means 1, 2, or 3 methyl groups are getting attached. So that can also have different implications. So based on the context, the effect that a particular modification will have can vary. And if you want further details of it, you can look into this area in not just that book, but any review article or anything that interests you. But what I basically wanted to bring to your attention was the fact that not just the compaction, but modification of both histones and of DNA can lead to controller genetic information at multiple levels. So keeping this in mind, we will move forward towards the next step, that the histone code is something that has also been proposed. It is not something that is very extensively validated, but what it basically suggests is that the histone code is a hypothesis that states that DNA transcription is largely regulated by post-translational modifications to these histone proteins. Through these mechanisms, a person's phenotype can change without changing the underlying genetic makeup that controls gene expression. So what this effectively is implying is that while what we mostly look at is the genetic code that is what you have present in the mRNA and what that is encoding at the level of proteins, which means what will the ribosome come and recognize and what amino acid it will incorporate based on the sequence that it is finding within the mRNA. But that is not all that is controlling a person's phenotype, and instead the histone that is present within that region and the kind of modification that it is carrying can also have an impact on the type of genotype and effectively phenotype that the individual will carry. So the histone code is also something that needs to be factored in, and it is a hypothesis that supports the same. However, it is not a very hard and fast rule. It is not something which you can say universally. Therefore, it is still considered to be a hypothesis and uh, not a law or not a very strong fact. So keeping this in mind, what we see at the bottom here are um, I wouldn't say exceptions, but two other types of structures that can also be found within the within a cell at different time points and because of different reasons. But yeah, apart from the simple or regular modifications that we are familiar with, we can also see the presence of lamp brush and polytene chromosomes. What you see at the top is the example of a lamp brush chromosome, where you can see that DNA is sort of looping out. So you have a scaffold in at the center, and from the scaffold, DNA is looping out and forming these sort of abnormal not necessarily abnormal, but somewhat different from the usual structures that we see for 30 nanometer fibers, etc. And they are forming these structures where you have the central scaffold, and from there, DNA is radiating out into these loops. And these lateral DNA loops are distinctive for what we understand to be the lamp brush chromosome. And apart from that, what you see at the bottom is the example of a polytene chromosome, which was something that was first identified in the Drosophila salivary gland. And there you can see that there is a fusion between multiple chromosomes, and the point of fusion has been identified as a chromocenter. And you can see that it has the left and right arms of chromosome 3. It has the right arm of chromosome 2, which has undergone asynapsis. It has some portions of the X chromosome. So it is a very diffused sort of structure that is present. And again, this is not like a regular or a normal event inside the cell. It is an anomaly at the end of the day because it is leading to a genetic fusion, but it has been observed in Drosophila. And it is another way in which chromosome or chromatin can be organized inside a cell. So keeping this in mind, we can see that these are the different ways in which you can find chromatin to be organized inside the cell. Next, what you see on the right, this has been taken from a review paper. I have cited it as well. It is an it gives you a consolidated view of the different types of histones that are present. What we talked about till now was just this certain group of histones, the core histones or the nucleosomal histones, as one might call them, or the canonical histones. This is also another word. Canonical means regular or the default histones that you are familiar with. So H2, H2B, H3, H4, and H1 also. But depending again on the spatiotemporal regulation of gene expression, there can be exchange of the canonical histones with the non-canonical histones or histone variants. Sorry. So what you see here, this wheel is a representative of the various events in which you can see the exchange of histones or the um, a sort of barter system, sort of an arrangement that is present, that what was initially within the DNA, that was the canonical histone, that has been removed, and instead a different type of histone has been taken in because it serves one specific type of job. So the different types of histones that you can see here, we'll look at each one of them depending on the context. First, with respect to chromosome integrity, at the centromere, instead of these canonical histones, you can see the presence of CNPA, which was mentioned in the video lectures as well by Professor Lalorea. You can go back to it for understanding how it is significant, why it is necessary, and what job does it serve. Why is it present at the centromere? So the CNPA, H3.3, H2AZ, these are usually present at the centromere. Towards the telomeres, you will find a different histone variant, which is macro H2A, that is towards the ends, and that is distinctive of regular chromosomes at the telomere ends. However, for the inactive X, again, this is a phenomenon you will only find in females because they have two copies of the X chromosome, of which one copy is heterochromatinized and inactivated. That is a whole other domain of study, the X inactivation, and why is it necessary, what job does it serve, so on and so forth. Dosage compensation and X inactivation involves very largely the presence of the macro H2A, and you can see that its presence is throughout the chromatin, not just towards the end, throughout the chromosome, not just towards the end, and that results in complete inactivation of the X chromosome. Next come the cell fate transitions. So depending on what condition that cell is currently in, which is changing its fate or which is allowing it to transition from one fate to another, there can, in a large number of cases, be exchange with other histone variants. So you can see that when there is reprogramming or differentiation, you can see there is exchange from the default histones to H3.3, macro H2A, H2AZ, H2AX, etc. Then when cells are going through EMT, that is epithelial mesenchymal transition, then as well you can see that there is a change in the cell fate where it was previously epithelial in nature, which means that it was bound to a substratum. And now it is transitioning to the mesenchymal state, which means that it is no longer bound to a substratum. It is free to move about. And this event is characteristic of cancer cells, where from the primary tumor site, they undergo some changes which allow them to migrate or which allows them to metastasize. Now, this phenomenon is known as EMT, or epithelial mesenchymal transition, and it involves certain types of histone variants as well, like we just talked about. And finally, during proliferation or senescence, when a cell is deciding that it is going into senescence, it is no longer going to proliferate, then as well you can see the involvement of certain types of histones, mainly macro H2A, H2AG, J, H3.3, and H2AZ. So these are the subtypes that are involved in proliferation and senescence. You don't need to remember all of these examples. Please keep a few of them in mind, mainly the ones which were given in the lecture notes by Professor Lalauria. But apart from that, if this is something that interests you, you can go to this review article. It has been beautifully explained. It might be slightly advanced, but it is an interesting read, so you can try it out. And finally, DNA damage response is also the key event or umbrella event under which you can find these histone variant exchange to occur. For example, for chromatin accessibility to be changed, you can find macro H2A and H2AZ involved. For DDR or DNA damage signaling and repair, you will find H2AX, H2AZ to be involved. And for transcriptional regulation also, you'll find the involvement of H2A and macro H2A. So these are the different variants that come in, and the presence of these variants can alter the interaction between DNA and histone. Their rate of exchange can also vary depending upon the type of histone variant that is coming in. It has a significant role to play, and therefore it can either localize at that location and stay there, or it can get exchanged depending on the type of condition that the cell is in, and that will lead to different cell fates, different regulations, differential chromosomal integrity, or the response to DNA damage, and so on and so forth. So, uh, so with this in mind, we will next move to the other proteins that are involved in chromatin organization and condensation. So again, if there are any doubts, please interrupt me. I might be going a little fast or might be giving too much information. Please interrupt me. Please ask any questions that you might have. We'll try to address them right now. If not, I will get back to you with more information. So please, please, please do let me know. Um, Coming back here, what you see is the image. It's an electron micrographic image, particularly it's a scanning electron micrograph, or SEM as it is commonly known, of human metaphase chromosomes that show paired identical chromatids that are associated along their length. But the association is, while it is present, it is slightly loose throughout the length of the chromosome. But at the centromere, it is very condensed. If you can see that, that is the primary constriction, that you can see that along the length they are associated, but there is a certain distance that is present there as well. But at the centromere, you can find a cinching or sort of a tightening that you can see at the primary constriction. And this is because of differential proteins that are operating at different positions within the mitotic chromosome as well. Uh, these chromatids or sister chromatids are not split apart from each other until anaphase comes in. And what you see here is the example of a metaphase chromosome where they have condensed to their maximum, but have yet not undergone the alignment or segregation processes. What you see on the right is a fluorescence micrograph of the metaphase chromosome in a cultured human cell. The DNA here is stained blue. What you see in green are the kinetochores, and cohesin, the protein that we have previously discussed to be extensively involved in this cohesion process, has been shown here in red. At this stage of mitosis, cohesin has been lost from the arms of the sister chromatids, but it is present at the centromeres where the two sister chromatids are very tightly joined. So under normal circumstances, you would find it to be present along the length of the sister chromatids as well, not as highly expressed as at the centromere, but it is definitely present in holding the two sister chromatids close to each other. Now, the protein that is responsible for this purpose, in order to keep this cohesion intact, are the SMC group of proteins. Again, this is something we have previously discussed as well, and it is there in the lecture notes also. So SMC, or the structural maintenance of chromosome, SMC group of proteins, are involved, as the name indicates, in maintaining the structural integrity of chromosomes throughout the different stages of the cell cycle. What you see are mainly SMC1 and 3. This is a sort of a schematic representation only, but what you see are two groups, or two representatives of the SMC group of proteins, along with a few other proteins as well. What they mainly involved is a head domain and an ATPase region, and interaction with a few other proteins like RAD21, SAWAPL, or PDS5, all of which together maintain this sort of locking system, and that lock goes and binds around DNA, and it holds that not just the DNA but the chromatin fiber that we are referring to. It binds around it and it holds them close together. It holds two sets of chromatin fibers close to each other and maintains the cohesion between the sister chromatids during these stages of the cell cycle. What you see, I think it has, yeah, it's okay. What you saw in the diagram only has been described here further. So the SMC group of proteins, they are conserved in eukaryotes as well as prokaryotes. They are not just responsible for maintaining the genome, but they are crucial for effectively for cell viability as well. So they are essential for the viability of yeast cells as well. Their orientation or their structural arrangement is such that they have one ATP binding head domain, they have flexible coil regions, and they have a hinge region. Why is the hinge region important? If you look at the diagram that we just showed here as well, that for this bending sort of structure to come in, they need to have a hinge. Apart from that, they have an ATPase domain, which is necessary because this process of cohesion or holding sister chromatids together is certainly an energy extensive process. And they are able to dimerize with another SMC partner to form a heterodimer at the hinge region. And the head domains are bridged by the non-SMC subunits, which we saw in the diagram as well, MCD1, SSC1, RAD21. In this case, it was RAD21 in budding yeast. And these non-SMC units are these SCC1, MCD1, RAD21, and SCC3 or SA proteins. You have other regulatory factors as well, such as WAP1 and PDS5, which were also there in the diagram. They are also often regulated by post-translational modifications of various subunits like acetylation, SMOylation, and phosphorylation. They associate with DNA, and they're involved in many DNA transactions because of their ability to bind and tether, which means they're able to bind and they're holding on to DNA, and they are maintaining the arrangement and the compaction of chromosomes throughout the different stages of cell cycle. So this is for the basic group of SMC proteins. Particularly when we talk about cohesin, cohesin's job, as the name indicates, it is to glue together or to keep cohesive forces intact between two sister chromatids. It is majorly involved in mitotic chromosome condensation. It also has roles to play in double-stranded break repair by homologous recombination. It has a role to play in DNA replication as well. It increases the speed at which replication occurs, so it is filling up the fork. It has its job in gene expression as well as acting as a silencing barrier. If that is, if a region has been condensed by cohesin, it will affect the genetic expression at that region or at that locus, and can act as a barrier or can act as a silencing factor as well, depending upon the condition that it is in at that point of time. Apart from cohesin, the second group of proteins that are majorly involved in this structural integrity are condensins. Where are they coming about? For your reference, you can look at the image on the right, and you can see that during interphase, when the chromatin is not that tightly looped or tightly organized, you will be able to see that cohesin is still present there between these two set of or two regions of homologous chromosomes. Cohesin is operating in that regard, and the structure of cohesin is shown on the in the inset picture as well, with SMC1 and SMC3. However, when the cell is entering prophase, where nuclear material needs to be, it needs to undergo some organization, and some nuclear compaction has to occur, the envelope will dissolve and the nucleus will now move to a direction that its genetic material or its chromatin needs to be condensed into chromosomes. And for that to be done, cohesion will not be enough. At that point, you need to loop DNA, you need to form the 11 from the 11 nanometer fiber, you need to form the 30, 300, and so on and so forth. Packaging of chromatin has to be performed, and for that to be carried out, you need to have condensins. Condensins are the second group of proteins. They have SMC2 and SMC4 instead of the ones that we saw for cohesins. And they have a similar structure though, because they are made up of SMC group of proteins only, but their job is different. They are condensing chromosomes, which is necessary only before or only when a cell is undergoing the division process. That is only during prophase when it needs to be condensed. So you can see that at the edges of the scaffold, or at the edge of the looping area, you can find condensins present. Between these two entities, or between these two copies of chromosomes where condensation is occurring, there is a cohesion that occurs even at this stage. Again, this cohesion is being maintained by cohesin. So just look at this diagram very carefully. It should be good enough for your reference. If you want to read up on this further, I have mentioned the book at the bottom. It is present in Cell Biology by Cooper. It's not given in very extensive detail, but yeah, you'll be able to get a basic idea of the job of cohesin and condensin, and how they are not the same. They have different jobs, they work at different stages, and they can either hold two sister chromatids together from their loops, or it can just hold two copies of chromosomes together as well. So depending upon where it is present and what is its job, you can find the expression of cohesin and condensin to vary and to play its own destined role in each case. So, with this in mind, we are coming towards an end of the orientation or the structural organization and maintenance of chromosomes that occurs within a cell under normal circumstances. But what we next look at, which is what the slide I showed, is to look at some basic techniques that are used to understand DNA protein interactions. Now, a question might come to your mind that why is it necessary? So everything that we have talked about today is centered around the interaction between a DNA and a protein. Now, this we have just said with respect to the arrangement or the organizational genome inside the cell. But that is not the end point. The entire process of gene expression is also so heavily dependent upon the interaction of DNA and proteins itself, right? So if cohesin and condensins are performing the job of holding the genetic material at one place at one point of time, then there are n number of transcription factors inside the cell that are only able to execute their activity because they bind to DNA. If a transcription factor does not bind, that region will never get recognized, will never get expressed, and nothing will happen from that point onwards. Not just that, apart from that, if you also look at regions which need to be silenced, this was about gene expression. If there is gene silencing that needs to be maintained, then that also needs to be carried out by some repressor, or by these chromatin remodelers, or even for the chromatin remodelers to bind, they are proteins which are binding to DNA. So the extent to which interaction between protein and DNA is being carried out inside the cell is huge. So what comes to mind is that there needs to be ways in which we can study them. Some of the most basic techniques, or in fact, the most basic technique that was used back in the day was the filter binding assay, which is what is present in the next slide. So what you see here is that it is first of all, it's a method of understanding whether a particular stretch of DNA and a particular protein interact with each other or not. So because this is a relatively more primitive technique, the way in which it was studied was by using radio-labeled DNA. So you basically allow DNA replication to proceed in a medium which carries some radioactive nucleotides. These are otherwise known as hot DNTPs. Hot means they are labeled, which means they are radioactive, versus the regular nucleotides that we are familiar with. They are cold, or they are just normal DNTPs. So when you have such hot DNTP present, you will be able to detect its presence by using certain techniques which allow for radioactivity detection. So when you have double-stranded DNA, which is the first part of this schematic, when you have a mix that has just double-stranded DNA, and you pass it through a nitrocellulose filter, it's like a membrane sort of a structure, like a if you have seen filter papers, basically, it is much like a filter paper. It is just made of one particular material known as nitrocellulose. So double-stranded DNA is able to pass through the pores of this nitrocellulose filter. So after you have passed it, you monitor how much radioactivity is there on the filter by something which is known as liquid scintillation counting. You can count the radioactivity which is retained in the filter. And what you would normally see is that no radioactivity would be present because double-stranded DNA would not be able to bind to filters. Now, if you take your protein of interest and you directly pass it through the membrane, there you will see that the protein is able to bind because it is much larger in size. It is not passing through the membrane, but it has to be labeled differently. For example, if your radioactive DNTP was labeled with say for example, hot phosphorus, if it was labeled with hot phosphorus, then your protein can be labeled with hot sulfur, or radioactively labeled sulfur, heavy sulfur. Alternatively, in today's time, we can perform these experiments at a much higher resolution. We can use fluorescently labeled DNTPs and amino acids instead of these. But of course, if you have these advanced techniques, you do not go through filter binding assay. But the principle of it is simple and easy to understand. When you have double-stranded DNTP, double-stranded DNA, it passes through the filter. You will not find any radioactivity. When you just have your protein, it is retained on the filter, but it will fluoresce green. The protein remains bound to the nitrocellulose membrane. When you have a mix which has both your radiolabeled DNA and your protein mixture also. So when you pass this DNA protein mixture through the nitrocellulose filter, you will be able to see, you will be able to detect radioactivity, and this detection would be different from that of the protein because while it was labeled red, in this case it is labeled green, and this double-stranded DNA, which was previously not able to bind, is now able to bind because it is interacting with the protein. In case you have some non-specific DNA sequences, if you use non-specific DNA sequences, then they would not be able to bind and they would be able to pass. So if your DNA and your protein of interest are interacting with each other, they would be retained on the filter and you would be able to detect the same. On the other hand, in case it is not interacting with each other, then the detection will not be on the filter, but instead your radioactivity would be present in the elute, which is the solution that has passed through the filter. So this was one of the most preliminary ways in which protein DNA interaction was studied. Now, why at all did I stress on this? Because this, even though this is one of the most preliminary techniques, and this is not something which probably most of us would perform in the lab today, or anybody who is hoping to work in a research lab in the future will even resort to. But this is one of the easiest ways. And if you are ever appearing for an interview for your own knowledge, you might be asked that if you don't have any resources, if you don't have any fancy machinery, and you want to know if your protein and your DNA of interest are able to bind to each other, this is the easiest way in which you can do so. So for simplifying techniques and for keeping a comprehensive view of how DNA and protein can interact with each other, this is one of the best techniques. So it would be good for you to keep in mind. The second technique that is often used is the gel mobility shift assay or the electrophoretic mobility shift assay, otherwise known as EMSA or GEMSA. This, as the name indicates, there is a shift in the mobility, electrophoretic mobility or gel mobility when there is an interaction between the DNA and the protein. So if you look at the schematic on the left, you can see that you have a DNA fragment initially, which is labeled. The yellow mark at the end indicates that there is a label attached to it. You have you split the sample into two parts. To one, you add your protein of interest or you incubate with your protein of interest, and the other, you do not add your protein. And then you subject it to electrophoresis. I hope you're all familiar with electrophoresis. If not, please go ahead and ask me. So basically, you look at the movement or the migration or electrophoretic mobility of either the DNA or the DNA protein complex through the gel. Now, under normal circumstances, what you see in the schematic on the right will represent what can happen. When you have bare DNA, which is not binding to your protein of interest, then that will migrate the fastest because its molecular weight is quite small, quite limited, and it is able to move through the pores of the gel that you have uh, taken in the experiment. It is able to migrate fast, and therefore you can find the band to be quite at the bottom or at a low molecular weight. Versus when you have a DNA protein complex, then under those circumstances, it will migrate much slowly because its molecular weight will now increase, and that DNA protein complex is not able to move through the gel quickly. And you can see that there is a shift in that case. You are able to see a band in the second lane, in lane number 2. You will see that there is a shift in the mobility through the gel. In the third lane, what you see is the presence of two proteins. Now, this is another circumstance where if you have more than one protein of interest, then if you have multiple proteins of interest and you want to see if both of them or all of them are interacting with the DNA or interacting with each other, whatever the case might be, then you can have multiple lanes in which you will study. And in case both of the proteins are interacting, you will see further shift in the movement, and that will indicate that yes, this is the order in which DNA protein interactions are indeed occurring inside the cell. The reason why this was so relevant is because this technique has been extensively used to understand how transcription occurs, eukaryotic transcription occurs, the order in which these proteins assemble with each other in order to form the transcriptional machinery that we are all familiar with at this point of time. So very basic technique, but very extensively used. What has only changed over time is that previously radioactive strategies were used. Now we use relatively safer non-carcinogenic methods to try and arrive at conclusions which are equally relevant. So with that, protein DNA interaction methods are complete. And finally, this slide is just to sum up what was presented in the last two lectures for this video for this week. So they talked about the 3C method, the chromosome compaction and conformation, chromosome conformation capture assay. So it involved these simple steps of cross-linking, digestion, ligation, and finally detection by PCR, and then plotting of the contact maps. And from the observation that was initially seen, what they concluded was that the presence of the loops or chromatin loops in an organism such as yeast is responsible for promoter enhancer interactions, and that is controlled by how close they are to each other within the same chromosome. The formation of a heterogeneous loop in individual yeast cells can be observed in population of cells with homogeneous gene expression. The presence of loops genome-wide in human cells, but only a subset of cells can be impacted by alterations in the loops. It means that not everything is present within the loop, but it can and the effect or the change which is occurring within the loop can affect a subset of genes and not necessarily all of the genes that are present in the loop. And the labs of Bendecker, etc., they had identified that the presence of these loops are not only for arrangement or only for compaction, but they have a very significant role in mediating DSB repair as well, or double strand repair as well. Because what you see here in the diagram on the right also indicated the same, that if a particular region is localized to a chromatin loop, then during the process of cell division, during meiosis, if there is a double strand break, then for homologous repair, the cell will preferentially use the template that is present closer to the loop and will try to correct errors within those parts and not use the one which is further apart, thereby quickening the process and making it more accurate and reducing the possibility of any duplication or deletion from coming in within this repair process. So their observations were quite remarkable. And yeah, I think, yeah, with this, I'm coming to an end for the topics that were part of the recap for this week. So majorly, I discussed the process of, yeah, I discussed the way in which chromatin organization is carried inside the cell, and what are the subtypes that come in of the histones, what are the variants, why are they relevant, where they can play a role, and the different groups of proteins that are operating, the cohesins and condensins, what is their job, and yeah, basically these were the topics that were part of this week. Again, if there are any doubts, please go ahead and ask, and I will share a few sample questions, and then we can wrap up the class for today. Yeah, I think the screen should be visible now. Mhm. So, this was from the previous year. Uh, Again, if there are any doubts, please do interrupt me. So the first question is about the nucleosome core particle. So each nucleosome core particle is separated from the next by A, histone tails, B, linker DNA, C, linker RNA, or D, SSP proteins. So of course, like we discussed, that in between the region or the stretch of DNA between two nucleosomes is known as the linker DNA, and separates one nucleosome from another. So the answer to this is linker DNA, and the rest of them are, of course, not correct. Second, the cleavage of MCD1 by separase. I did not discuss this today, but I think it was present in week 4 interaction probably. That what is the job of separase, how it acts, and it leaves, it allows the separation of the sister chromatids during anaphase. So you can go back to that lecture if it is of interest to you. So the cleavage of MCD1, which is present between the sister chromatids, are present as part of the complex formed by SMC proteins. That segregation or that cleavage by separase triggers the movement of sister chromatids in what direction? The options I've talked about, either opposite halves or a single half of a cell. Of course, it is towards opposite halves, which is what we are familiar with. And this occurs during anaphase. Therefore, the correct option is option A, that separase is allowing the movement, or the cleavage of MCD1 by separase is allowing the movement of sister chromatids into opposite halves of the cell during anaphase. So that is the correct option. Then coming to question 3, the condensation of chromosomes during mitosis is essential because of which of these reasons? So A, it prevents the entanglement of chromosomes with each other and cleavage of DNA during cytokinesis. Second, it enhances the efficiency of replication. It prevents cells from entering meiosis, or none of the above. So, of course, condensation of chromosomes is necessary for the process of meiosis to proceed smoothly, and that is being ensured by preventing the entangling of chromosomes with each other, or maintaining the chromosomes in an organized manner, and for cleaving them during cytokinesis, preventing their cleavage during cytokinesis. So, option A is correct. The rest are not true. It does not affect replication, because replication occurs a lot before that, and replication does not require condensation of chromosomes, and it is something which is occurring during prophase. And it does not prevent cells from entering meiosis as well, because condensation does not occur before that part. So the correct option is option A. Then in question 3, the correct statement about TADs, which are the topologically accessible, like the domains within chromatin where they are being arranged in a certain manner, topologically associated domains. So which of them are the correct statements about TADs? A, that they are topologically associated domains. Yes, that is the expanded form of this abbreviation. They are present in interphase chromosomes. Yes, that is also true. Their formation depends upon SMC complexes. Certainly, they are responsible for forming the loops which are necessary for TAG domains. And therefore, the correct option is option 4, all of the above. Then for the next question, the histone code hypothesis, which I discussed in the interaction session, essentially states that A, histone modification is important for the regulation of cellular function. Each combination of histone modifications would attract specific proteins that would then execute the appropriate function. Specific modifications on the histone tails provide stability to the nucleosome complex, or none of the above. So among these, even though the statement that I showed is not directly being represented here, what you need to understand is the consequence of that statement and consequence of what histone code means. It basically means that apart from the code that the from the genetic code that we are familiar with, which is necessary for translation for the ribosome, the presence of a code on the histone, and that code is not just limited to sequence, but that is limited to histone modifications as well, that can call or that can recruit other proteins that execute their function and that control whether that region of the genome will undergo expression or not. And therefore, they can effectively affect the phenotype of that person, even though the genotype of that person is not getting affected. So it bridges the statement that I have shown and what is being asked here. And therefore, the correct option would be option 2, that histone modifications are attracting specific proteins to execute their appropriate function. Then the nucleosome core particle consists of which of these following? So we have discussed this extensively. You have two molecules of H2A, H2B, H3, and H4, along with 147 nucleotide base pairs, which are wrapped around the double-stranded DNA. And therefore, that would be the correct option. Um, Phosphorylation of histone tails occurs at which of these amino acid residues? I did not discuss phosphorylation, but I think it was present in the lecture notes. And in general, within the system, majorly phosphorylation occurs only at serine and threonine. Majorly, there are exceptions to this. But yeah, serine threonine phosphorylation can occur very frequently. So the correct option in this case is serine. Then, what are the main experimental outcomes of the double tethered DNA assay, which was a part of the fourth lecture? A, to understand the kinetics of DNA binding and modifying protein or protein complex, to understand the loop extrusion properties of the modifying protein or protein complex, to understand the directional properties of the loop extrusion by the modifying complex. So technically all three of them have been studied. So they looked at how DNA binds to the how DNA is bound by protein or by the protein complex, how the looping is being carried out. When we say kinetics, it means the rate at which it was occurring, how the loop was formed and not formed. If you remember the videos that were shown as part of the micrographic videos that were shown as part of that lecture. And also they had introduced a buffer flow to show that the direction of loop extrusion can also be monitored using this assay. So all of these conclusions could be drawn from the double tethered DNA assay. Then what is the correct chronological order of steps that is performed during the micro-C or the C3 experiment? So I did show that and it was part of the video lectures as well. So first of all, you need to cross-link the nucleosomes in order to conserve interactions as it is. Then you need to perform chromatin digestion by MNase or micrococcal nuclease. After digestion has occurred, you will allow ligation to proceed. So only those loops which are close by would be able to patch up with each other or would be able to ligate with each other. And then you purify it and then you sequence those regions to understand which areas are part of these loops, of these condensed loops. So based on this sequence, the correct order would be first one, that you have cross-linking nucleosomes followed by digestion, ligation, purification, and paired end reading. So that is the correct option. And I think this is the last question. Yeah. Select the correct outcome of the micro-C technique by Professor Koshland. So first one is that the base of the loops, they correlate with the occupancy of the cars. There is only one type of bound cohesion. So the first option is correct, that how the occupancy of the cars is what is showing what is the base where the loops are being formed based on the contact maps. There is only one type of bound cohesion population, that is static. This is not true. If you remember, there were two types of populations. It is present in the slide I showed as well and in the lecture notes as well. So there are two types of cohesions, those which are static, those which are dynamic, and the presence of these or the differential presence of this is what leads to the varied contact map that people observe, because when these experiments are performed, they are not performed with a single cell, usually not performed with a single cell. You have a population of cells, and within different populations, you will find the different differential binding of cohesions as well. And cohesions stably bind to cars prior to loop formation, and this is of course true. So both statements 1 and 3 are true. Therefore, option B would be correct, and rest of the options are not. So yeah, I think with this will be done. So with this, I think we have arrived at the end of today's class. If there are any questions, please go ahead and ask. We basically looked at some sample questions in this regard. I did not get into all of the topics, like I mentioned previously as well. But the idea was to highlight the significance of basic biology research, which showed how epigenetic modifications are occurring inside a cell, how the chromatin is packaged, what is the implication of it, what is the need for it, and how the presence of this or the understanding of this chromatin organization is being used today to understand this chromatin control and genetic expression control at a much larger level, at a much more advanced level, using all of the techniques such as 3C and the microscopic techniques, etc., to understand how genetic regulation is taking place and how chromatin organization is helping in that regard. So with this, I think I have conveyed my end of things. If there are any questions, you can please go ahead and ask them now. I'll just turn off my camera. If there are no questions right now, it's okay, not a problem. You can bring them up in the later classes, and preferably, if you want, you can make a list of these questions and we can discuss them in the last extra session. I think it is on 19th. I'll just look at the calendar once. Yeah, on 19th, there will be a session from 6 to 8. It's an extra session, not a official weekly session, where we can discuss any and every doubt that you might have had during the tenure of this course. And if you want any additional references or anything that you wish to be clarified, we can discuss those in that class. Apart from that, if there are any doubts, please do post them on the discussion forum, and somebody will get back to you with an update on it. And yeah, so thank you so much for joining today. The next session will be on Sunday from 4 to 6 for discussion for the topics for week 7. And then we'll have another session for week 8. And finally, we'll complete the entire course with an extra interaction session on that day. So I'm hoping these sessions have been helpful to you. Please come up with questions if you have any, and please let me know if there are any topics that you want me to clarify further. So thank you so much for joining, and I hope you have a good day. Yeah, Deepti, please go ahead. Deepti, you have a question?
Hello.
Yeah, I can hear you.
Good evening. Hear me, ma'am?
Yeah, yeah, go ahead.
Yeah, actually, the sessions
Your voice
sessions
is breaking,
are
Deepti.
happening. Hello.
Yeah, now
Hello.
I can hear you. Yeah.
Yeah. So, do we need to attend all the sessions? Means it's not possible to attend all the session. Means I am following your sessions. So rest other teachers are also means professors are giving the sessions. So do we need to follow all the sessions?
That is up to you. Even if you can't attend all the live interaction sessions, you can go through their slides. That will certainly
Hmm.
be helpful to you. And you'll find their videos. I think you have that spreadsheet most probably with everybody's YouTube links. So it is possible that you might find something useful in someone else's lectures as well. But if that is not feasible, you can go through the slides of all of the everybody who is hosting a live interaction session. And from there, you can, if you find some part which is not very clear to you from the slide directly, you can go back to those lectures. But attend as many sessions as possible for you. It can be helpful, but yeah, it's not a compulsion, of course not.
Okay, the topics which were covered in all the sessions are same, right? The four,
Yeah, yeah.
fifth or
It's
sixth
about different
topics are
timings. Like something might, like
yeah.
a Saturday or a Sunday might be convenient for some students. For some, it might be a Monday. It's up to whoever is like comfortable on whichever day.
Okay, the topics are same, right? That I'm
Yeah, yeah.
doing. I'm
Somebody
seeing.
might come up with extra questions. That is why I'm asking you to go through the slides. That someone might have other questions that can be helpful for you. But yeah, the overall idea is to cover the topics of each of those weeks. So you go through those weekly topics.
Okay. Okay. Thank you so much. Thank you. Bye.
No worries. Okay. Thank you so much for joining and good night.
Thank you, ma'am.